Okay, welcome back, and today we're going to talk about Chapter 3, and Chapter 3 goes over the microscopy and methods that are used to enable us to study microbes and microorganisms visually. And so we're usually talking about looking at one or a few microorganisms at a time and looking at their what we call their microscopic morphology, or their appearance under the microscope. So far we've gone over kind of a general overview of microbiology, a little bit of history, how we know microbes exist despite that we can't see them. And really microscopy was fundamental in the birth of this field of biology because we just couldn't see these tiny little organisms before. So now today we can see them with the aid of microscopes, or we can measure or study them in indirect ways. And we've also gone over Chapter 2, which talks about infectious diseases, and so puts microbes in the context of human health and disease. All right, so this chapter I want you to be sure that you understand size. And of course in science we're talking about the metric system when we're talking about measuring anything. And that includes the size of microbes. I'm going to tell you mostly about light microscopy and electron microscopy and how they're really useful in viewing microbes and microorganisms. But I'll also touch upon staining, especially gram staining, a really fundamental a tool that was developed around 150 years ago, and we still use in hospital labs today to help physicians make choices about treatment of bacterial infections. So let's get started. In addition to these objectives that I want you to learn as you go through the reading and listen to the lecture, I also want you to be aware that what we do in lab is also supporting what you're learning. So we're going to do a gram stain. We're going to use the microscope. You should be familiar with metric measurements, converting metric measurements of length, and also how microbes fall on this scale, a metric scale of length, and be able to calculate the total magnification when using a light microscope with varying objective lenses. Here's a brief summary of the terms that you should know. You should also look at the terms in the back of the chapter. But just note that I'm not going to spend time on some of the aspects of the chapter that go over in more detail other types of staining, such as fluorescence staining, and so pay attention to what I'm talking about here in the lecture, and be sure that you're learning the material according to the learning objectives that I've just shown. Okay, so we know that by definition microbiology is studying very tiny organisms. And those microbes, when we measure them, are on the scale of micrometers. So 1 meter is 0.00001 micron. So 1 times 10 to the negative 6th micrometers would be the measurement of 1 meter would be 10 to the 6th power micrometers. So they're very, very, very tiny units of measurement. Millimeters is 1,000 times. Millimeters is 1,000 times smaller than a meter, and micrometers is 1,000 times smaller than a millimeter. In the micro, it's abbreviated with this kind of U-shaped letter that's the symbol for micron. So, just looking at some examples from your book, we have viruses, which are the very smallest of the microbes. Viruses, viroids, and prions would be much smaller than, say, bacteria or fungi. So here we have the herpes virus at 0.2 micrometers. You should also be familiar with the next unit down from micrometers, which is 1,000 times smaller than a micrometer, which is a nanometer. And so 0.2 micrometers would be the same as 200 nanometers. Bacteria generally range around 1 micron. Staphylococcus aureus is spherical. It's just under 1 micrometer in diameter. Escherichia coli is rod-shaped, and so it's a little longer, but the width is about 1 micrometer. And then there are some bigger bacteria. Bacillus megaterium, as its name implies, is 4 microns, but generally you're talking about bacteria being about 1 to 2 micrometers or microns in length. Just for comparison, a human red blood cell is 8 micrometers. Eukaryotic microorganisms like paramecium, which is a kind of amoeba, a protozoan, can be as large as 100 micrometers, but generally unicellular microorganisms such as yeast, which is a type of fungus, are only a little bit bigger than bacteria. So again, in the range of a few micrometers. So microscopy helps us to view these microbes and microorganisms, and that's how we're going to use the microscope in lab in order to do such activity. And really the way that a microscope helps us to see such tiny organisms is that it has 3 variables. First, it makes things look bigger, so it magnifies them. It also has the ability to help us to distinguish between 2 objects that are very close to each other, so gives us the resolution that we need in order to be able to see objects as 2 separate entities. And contrast also helps us successfully see microorganisms under the microscope. And I'll come to that later when I talk about staining, because staining is really what improves the contrast enough so that we can easily see bacteria under the microscope. So just back to the scale. As I mentioned, we used metric units of measurement, as always in any science course. And here are some more examples. Down here at the very smallest end with about 1 nanometer in size, you're talking about DNA. So prions, these infectious proteins that cause gradually progressing neurodegenerative disease, would be down here on the scale of size in the range of nanometers. There are a fairly large type of virus that infect bacteria called bacteriophages. They have a rather complex structure when you're comparing it with other viruses, and they are up around 100 nanometers, 150 or 120 nanometers. And that is about tenfold less than the typical bacteria. Here we can see a scanning electron micrograph giving this kind of surface view of the three-dimensional shape of Escherichia coli. And then again, red blood cells around 8 micrometers. So keep that in mind as the range, and just remember that eukaryotic microorganisms that are unicellular, such as yeast, fall somewhere in between the E. coli shown here and the red blood cells. So we can see some microbes, so actually microorganisms may be not the most accurate term, but these are very, very small organisms that most of which we need some sort of microscope in order to see. Certainly bacteria we can use light microscopy for, because the lenses, especially for a compound microscope where you're using 2 lenses to get a greater magnification, we can easily see bacteria and yeast and other unicellular microorganisms. But when you're talking about the inner details of a bacterium or a yeast, or when you're talking about looking at viruses, you really need to get much more powerful magnification, which usually means using an electron microscope. So either looking at the surface of an image using scanning, a surface of a specimen using scanning, electron microscopy, or looking inside at the more internal details of a microorganism or a microbe, such as a virus, by using transmission electron microscopy. Finally, when you want to get down to the molecular level, you really need to use other techniques to look indirectly and determine the size and the shape indirectly, such as using techniques like X-ray crystallography, which can tell us the shape of a protein here, in this image E, or more details about molecules like ribosomes and RNA, shown here in this transmission electron micrograph in image D. So, the things that influence our ability to see microorganisms using microscopy, are especially contrast. So naturally, microbes like bacteria really have very little substance and don't have color. So being that they're transparent, we use staining to add contrast, or we use special kinds of phase contrast microscopy, special kinds of light microscopy, in order to see them in more detail and more easily. The wavelength of light has to be smaller than the object we want to see, and that's why sometimes, it's necessary to use an electron beam rather than a light beam in order to see smaller details, internal details of microorganisms like bacteria. So the wavelength has to be smaller than the specimen, and in the case of an electron beam, the wavelength is smaller than a wavelength of light would be. And then you need some sort of detector with sufficient ability to resolve the wavelengths that are hitting it. So the properties of light include absorption, reflection, refraction, and scattering, which are defined here, and these all are important you have to control all of these behaviors of light in order to optimize the amount of light that gets to the eye that's observing the specimen. So for example, in the light microscopy that we use, we're going to have light that's going from the source at the bottom of the light microscope through a glass slide, and then through the air, and then through a glass of the objective lens, and then through the glass of the ocular lens to our eye. So when the light goes from air to glass, because the refractive index, or the measurement of refraction of that material, is different in air versus the glass, the light will bend when it comes through that when it goes through different materials of different refractive indexes. So in order to minimize that bending, because we don't want it to the light to bend so much that it no longer goes through the objective lens, because then it wouldn't get to our eye, and it basically ends up making the image seem fuzzy when we look at it. And so we want to minimize the bending so that the focal point of the light continues through that objective lens so that we can see everything clearly. In order to do that, we add a material that has a very similar refractive index to glass, and that's the immersion oil that we use. Now we put a drop of that, slide the objective lens into place, and that way the image will be crisp and clear. So the focal point of a small object when it reaches the lens can be used to make the image appear much bigger, or the specimen on that object that we're looking at appear much bigger than it actually is. And when we look at a single lens, when it's biconcave, or biconvex, excuse me, biconvex, then it will flip the image upside down and magnify it at the same time. And as we learned about in Chapter 1, it was really Francisco Redi that was very good at grinding these lenses and got a greater magnification than his predecessors, and was able to really see a lot of detail in the specimens that he looked at. So if you have poor resolution, you'll get a kind of fuzzy image like you see in this circle down here in Part B. But if you have good resolution, you'll really be able to see the crisp details as seen in Part A. And that certainly what you see in Part A, you can see that two separate objects in the specimen can be easily resolved. Whereas you're not really too sure in Part B because they're unresolved. You can't. The resolution is because of the wavelength that's hitting the object, the resolution is, and also because of the bending of the light, possibly resulting in less light going through to the detector. So again, this is why we use oil when we use the oil immersion lens. And you have to be sure that when you use the oil, you put a big enough drop, just one drop, but a nice large drop, so that it actually makes this connection between the glass of the slide and the glass of the objective lens. That way you will have much less refraction of the light, much less bending, here seen here with the blue line, rather than if you just had no oil, which is depicted by the red line, and you can see that the light is getting lost and will never be some of it gets lost and will not be seen, so the image will appear fuzzy. So you should be familiar with the parts of the compound microscope. We're going to do this as an exercise in lab, so I'm not going to spend a lot of time talking about it. And you should review it on your own before coming to lab. But I do want to review how to calculate some simple math that is used routinely when doing light microscopy so that you can determine how much you are magnifying the object you're looking at. Okay, so in the microscopes that we use in the lab, they're light microscopes and they're compound light microscopes, which means that they're using more than one lens, and in this case we're using two lenses. We have the ocular lens, which remains fixed. We can't change the magnification of that lens. It's always 10x. But the objective lenses, we have four different objective lenses: the 4x, which we call the scanning objective lens; the 10x, which is the low dry objective lens; the 40x, which is the high dry objective lens; and then the oil immersion lens, which is the 100x. And the way that we detect how big the microscope is how much the object is being magnified by the microscope is just by multiplying. So 10 times 100 and you get 1,000. So really, for our purposes this semester, we will not be able to see any bacteria until we get to that oil immersion lens, which will give us 1,000 times magnification of the actual size of the bacteria we're studying. In addition to magnifying the object, it's important to add contrast to the microbes because, as I mentioned before, they are colorless, and so if you put them on a clear glass slide, it's going to be really hard to find bacteria without giving them some kind of color. So you can do that by just simply adding a stain, washing, and viewing the bacteria, or in the case of the gram stain, which we'll be doing, you can use more than one stain to see how it behaves differently in different kinds of organisms. And so the differential stain allows us to distinguish between the two largest groups of bacteria, which is medically relevant for the physician to choose treatment of bacterial infections. So there are two major groups of bacteria are gram-negative bacteria and gram-positive bacteria, and they stain differently by the end of the gram staining process. The gram-negative will appear red, while the gram-positive will appear purple. And those are based on differences in their cell wall structure, so fundamental differences in their structure, which also reflects their susceptibility to antibiotic drugs. Now we'll be doing this in lab, so I'm not going to go over the details of these steps. You can read about it in your textbook. But I do want to mention a few things. One is that in order to stain, you first need to make sure that the bacteria are attached to the slide. And in order to do that, we use heat fixation. So we draw it through a flame very quickly, 3 times. After staining, you have to wash really well so that you don't have any crystals of stain on the slide. That will be about the same size as bacteria, and so it will make it hard to find the bacteria on the slide. The gram staining method is important in distinguishing, as I mentioned, these two major groups, the gram-positive cells and the gram-negative bacterial cells. And the reason it's different is that the gram-positive cells retain this first stain that's used, crystal violet, throughout the whole process, whereas the gram-negative ones have a different cell structure that when the ethanol or decolorizer is added, it loses that crystal violet. And that's because the additional layer beyond the cell wall, beyond the peptidoglycan of the cell wall, is a lipid bilayer consisting of lipopolysaccharide and phospholipids, and that is really just destroyed by the addition of ethanol, and so the dye just seeps out during this decolorizing step. And then we would have a awfully hard time seeing the gram-negative cells unless we added another color that's different than the crystal violet color, and so we use safranin, which is a red stain, in order to view those bacteria once the destaining or the decolorizing step is complete. So here are a couple of actual examples of light microscope images of gram-negative cells. Here you can see in the case of, and these are switched actually, the gram-negative cells are on the bottom here, some rod-shaped gram-negative cells, and the gram-positive cells are on the top, along with some red-shaped eukaryotic cells, maybe human cells if they were, if it was a sample from the mouth or the gums, for example. These almost round, very short cells are probably Streptococcus species, and they retain the purple color and are gram-positive. So gram staining is really a key diagnostic tool. It's used routinely in hospital labs every day, and that's because of these differences in the cell wall that then predict whether the bacteria would be susceptible or resistant to antibiotics that the physician has to choose from for treating the infection. One thing about staining, another tip about staining is that you really only, it really only works well when you have live bacteria. If the cell wall and the integrity of the cell is breaking down because the cells are dying, then you will get confusing results because you may see both red and purple in your staining. And of course you have to have just one type of bacteria present that you're studying, otherwise you won't know which result. You may get, again, two colors and not know which result applies to the bacteria of interest, to the pathogen that you're studying. There are other stains because not every bacterium gram stains. So, for example, Mycobacterium, in addition to their cell wall, they have this very resistant waxy coat of mycolic acids on the very outside of their surface, and it's very hard for really anything to penetrate that mycolic acid layer, and so special staining techniques involving steps where acid has to be added in order to facilitate the kind of open up this mycolic acid and break it down a little bit so that the stain can actually get into the cell and we can actually see those mycobacterium. Here in the light microscope image on the right are the mycobacterium examples here in this kind of deep red. And these, just one note, side note, these are actually several cells all in a chain, so all lined up. And the chain is what we call strepto in describing the morphology, not to be confused with the species Streptococcus, which it does, as its name implies, its shape, but really it's also used to describe morphology of other bacteria. And then there are special stains for special circumstances. So some gram-positive cells from the genus Bacillus, so all of the members of the species within the genus Bacillus and all of the species within the genus Clostridium, two types of gram-positive bacteria, will stain, if you stain gram stain them will stain purple as expected, but you may see kind of blank gaps at points along the cell structure, and those are where likely to be where spores are forming. Spores are very resistant dormant forms of bacteria. These bacteria, many Bacillus bacteria, are soil bacteria, as well as some Clostridium species, and in order to kind of survive in bad times when it's either very hot or very dry, the bacteria will go into a dormant state, and then when the good conditions return, they will then return to their the bacterial cell form that can grow actively and reproduce. So, but these spores are very resistant. Again, they don't allow anything to penetrate their surface, and so in order to stain them, you have to heat the endospores, heat the sample that has endospores, and then you can see this kind of turquoise color here within the pink of the stain, which we call malachite green, is showing where the spores are in the specimen. And the pink color around them is the cell that is producing the spores. So you can see that some of them have a lot of pink around them and the mother cell is still intact, but some of them are just the spore by itself, and the mother cell, once it forms a spore, will die. And that for those spores, they're just dormant spores, and they no longer have the mother cell surrounding them. And finally, in order to see some of these outside layers that are often virulence factors for pathogenic bacteria, like capsules that protect pathogenic bacteria from our immune cells, and so in order to study whether the bacteria have capsules, we need to do a special kind of stain called a negative staining, where we actually, rather than stain the bacteria, the goal is to stain around the bacteria. And here in the light microscope image, you can see that the bacteria are partially stained, so the internal part of the bacteria is stained pink. Then there's these big blank spots that are actually still part of the bacteria. It's outside of the cell membrane and the cell wall. They have these polysaccharide-rich coats, often called a glycocalyx or a sugar coat. You can think of it like the candy coat on an M&M. That surrounds the entire cell and is protective. And then all of the pink around that is just the background on the slide that's being stained, so that we can see that clear capsule. So light microscopy has limitations because the wavelength of light is too big to be able to get the resolving power or the resolution we need to see the internal details of bacteria and other microorganisms. And it's not even possible to view viruses at all using light microscopy. So instead, microbiologists use electron microscopes, which use beams of electrons, which are shorter wavelength than light. And that gives them a better resolution, even 1,000-fold greater resolution than microscopy. So in the case of transmission electron microscopy, which really is the most robust in giving the best resolution, you can get about 10,000 times the actual size of the image to 100,000 times the actual size of the image in magnification. And you can resolve two objects that are 2.5 nanometers apart. Okay, so remember that I gave you an example of a virus. I think it was a herpes virus that was around 200 nanometers, or a bacteriophage about 200 nanometers. Poliovirus is probably one of the smallest viruses. It's around 40 nanometers. So easily viewing viruses and their some of their internal details, and certainly easily viewing bacteria and their internal details. Scanning electron microscopy also gives better resolution than light microscopy, going up to 10,000 times the size of the actual image, object or specimen. Not quite as good as certainly, about tenfold less magnification than the transmission electron microscopy, but still gives good magnification and quite good resolution, but won't show you the internal details that you can see with transmission electron microscopy. Here's some examples. On the upper image, there are some virus particles that are budding from a white blood cell that it had infected, and all kinds of electron micrograph, so that the image that you get from an electron microscope are always in grayscale. But in the case of scanning electron micrographs, the computer is used to add color. So here you can see the difference between, in the image below, you can see the difference between the green objects, which are the bacteria, Helicobacter pylori, which is something that we carry normally, many of us carry normally in our stomach, and these are the gastric cells of the stomach, of the lining of the stomach, colored in this kind of tan color. So light microscopy has its limitations, but one thing that you can do with light microscopy, if you don't stain the microorganism, is that you can study live microorganisms. And that's not possible when you stain. You have to heat fix, which kills the bacteria or other microorganisms, and you have to kill because you have to freeze the specimen before doing electron microscopy, either scanning or transmission electron microscopy. So those are dead microorganisms or microbial agents that you're looking at. Here's the internal details of a paramecium that have been colored using a computer. And here's the surface image of a group of Bacillus-shaped bacteria. And again, you can see that. Here in the lower right corner, you can see the scale is listed. So this yellow bar is 2 micrometers. Here the yellow bar is 10 micrometers for the light microscopy. So you're getting much more detail seen with the scanning electron micrograph. And then here you have a bar of 15 micrometers, which is even more internal detail that you can see using transmission electron microscopy. Finally, I just wanted to touch upon a new form of microscopy that's called scanning acoustic microscopy that uses sound waves instead of electron beam to study living biological systems, and this is really useful for understanding biofilm, which is a community of bacteria and are particularly challenging problems when they are associated with infection. So this image shows is from a scanning acoustic microscope, and it shows the three-dimensional shape of the biofilm growth, in which these towers of many different types of bacteria form. And this is a living specimen. So sometimes when you kill the specimen, when you stain it, it can modify the actual appearance. And so by studying living organisms, you're really seeing how it really looks. Finally, as usual, I have some practice problems that I've chosen for you, so certainly take the time to check these out, as well as use the study guide in your preparation for our first exam.